So today I'll be talking about a manuscript that was published not too long ago, last month at the beginning of October, and the paper's title is The Translational Development of a Novel BAFFR CAR-T Targeting Therapy Targeting B-Cell Lymphoid Malignancies. I had the privilege to work under the laboratory of Dr. Hong Chen. Dr. Hong Chen is a MD PhD. He was a hematology oncologist for about a couple of years before he decided to become a PI to find new treatment options for leukemia and chronic lymphocytic leukemia. And so before we start off, this paper was published in Cancer Immunology, Immunotherapy, and part of the Springer Nature publication in 2023. And so what is a CAR-T cell, right? CAR-T cell is a chimeric antigen receptor T-cell therapy. This is a form of adoptive cellular therapy where you take cells from the patient, engineer them, and reinfuse them back into the patient. It's quite literally sometimes called a living drugs, because as long as these T cells are proliferating and circulating through your periphery, they will kill any cancers as long as they're living within your body. So this drug's, I guess you could say, half-life is the life of the T cell duration, right? So CAR-T cell therapy is a process of taking your T cells from the patient and modulating them in the lab, giving them a gene called CAR, which essentially normal T cells, they don't recognize cancers. They have the blindfold on. What the CAR gene does is it removes this blindfold and allows these T cells to then recognize the cancer and elicit their cytotoxic response and eliminate the cancer. So once we modulate these CAR T cells, they grow into millions of CAR T cells ex vivo, and we reinfuse it back into the patient, hoping for a robust immune response and eventually eradicate the tumor cells. And these CAR T cells, they are able to combine the properties of a normal B cell and also keep the T cell functionality. So to understand this, we have to understand a bit about the T cell physiology. So how does the CAR T cell differ from a normal T cell receptor? So if you take a look at panel A, a T cell receptor is MHC dependent. Inside our body, we have antigen presenting cells, which are usually our macrophages, our B cells, and our dendritic cells. And they have this molecule called the major histocompatibility complex, which digests proteins into peptides and presents it to our T cell receptor. However, this alone is not enough to activate our T cell. We need two signals. We need our co-stimulatory domain to activate our T cell. However, how a CAR T cell differs is that they use an antibody, the part of the B cell. They use a antigen recognition domain, and instead of recognizing a small digested peptide, they're able to recognize a target antigen directly on the tumor cell. And because they're MHC independent, they can overcome problems such as MHC downregulation in cancer, as well as co-stimulatory domain downregulations. And again, binding of this antigen to the CAR will stimulate signals 1 and 2 to activate the CAR T cells. So CAR T cells have been around for about the past decade, and there has been a lot of modulations of CAR T cells throughout the years. And what we call is different generations of CAR T cells. So the first generation, when the first CAR T came out, they had your antigen recognition domain right here, and they had your intracellular activation domain of a T cell. Now, the problem is that these CAR T cells, the first generations, they had trouble expanding in vivo inside patients, and it didn't expand well. So what they did in the next generation is say, let's copy the T cell receptor. Let's activate them in the same way that the T cell receptor is normally activated. Let's add a co-stimulatory domain. And that's what they did. And when they added the co-stimulatory domain in the second generation CAR T cells, they saw that these T cells were able to expand and proliferate within the body quite well. And then third generation is where you incorporate two co-stimulatory domains. And when you incorporate two co-stimulatory domains, what you normally see is increased persistence and expansion of CAR T cells within the patient's bodies. And a fourth generation is essentially one co-stimulatory domain with an added transgene. This transgenes can be increased cytokine production within the cell. It could be an effect that's normally not within the cell. So CAR-T cells have been really successful this past decade in treating ALL, and this is one of the diseases we'll be talking about today. So acute lymphoblastic leukemia. This is the disease of the hematopoiesis pathway, specifically the lymphoid pathway, where you have this overaccumulation of lymphoblasts within your bone marrow. And this can cause a number of problems. This will overcrowd your other normal cells. So it can cause anemia because it's overcrowding your red blood cells, or thrombocytopenia, right? It can lead to more easily bruising and bleeding, as well as nerve pain, right? You have these accumulation of blast cells within your bone marrow causing this immense pressure within the bone marrow, leading to nerve pain and risk of infection, because these cells are not fully differentiating. They're staying in their blast form. So you have a more higher risk of infection. So this is a picture on the left of a bone marrow tissue biopsy. And you can see in purple, these are your blast cells. And in a patient, a diseased individual, you see this overaccumulation of blast cells, right? So CAR-T has been revolutionary in treating relapsed ALL. And so now I want to talk about one of the first pediatric patients that was enrolled into a phase 1 clinical trial. And so this is Emily Whitehead. In 2010, she was diagnosed with acute lymphoblastic leukemia, and she went through the standard treatment regimen of chemotherapy, radiation, stem cell transplant, and targeted therapy. However, normally ALL has a high prognosis rate within children with 85 to 90%. She relapsed in 2 years. In 2012, she relapsed. And normally, if you relapse from the previous standard regimen, you become resistant. And at that time, if these standard treatment of ALL didn't work, that's essentially a death sentence. However, her parents did not give up hope, and they enrolled her into a phase 1 clinical trial in Philadelphia's Children's Hospital in the laboratory of Dr. Carl June. And she was the first pediatric patient in the world to receive CAR-T therapy. Now, at the time, there's been cases of this working in adults, but never pediatrics. So this was quite groundbreaking. And well, as you can see, she is still alive and well today, and she's a beacon of hope that CAR-T therapy to this day can still save people's lives. It worked extremely well on her, actually. It worked a little too well. When they had her second infusion, she underwent fever. She had really high fever. She had a cytokine, what we call in the clinical setting, cytokine release syndrome. She had elevated amounts of IL-6. However, during the clinical trial, because these T cells are rapidly expanding upon binding to the cancer cells, they're expanding so much, but they gave her a IL-6 antibody to help control the CRS. And to this day, she is still living cancer-free. And so in 2017, 5 years after Emily's clinical trial, the first FDA-approved CAR-T therapy was approved. This was Kymriah, or tisagenlecleucel. This targets CD19, and currently there are 6 FDA-approved CAR-T therapies. So there are 4 targets targeting CD19 and 2 targeting BCMA. BCMA is the B-cell maturation antigen, which is present on your plasma cells. So this is a good marker for your multiple myeloma cases. And if you take a look at the patient population, they all treat patients with relapsed or refractory disease. So CAR-T therapy is your last line of defense if your prior treatments failed. If the prior chemotherapy, radiation, and stem cell transplant failed, CAR-T therapy is there. However, we see so much success in non-Hodgkin lymphomas and acute lymphoblastic leukemia, but when we move over to chronic lymphocytic leukemia, we don't see that great of success. So if you take a look at this panel on the left here, we took targeting CD19 with CAR-T therapy, you have a complete remission rate of around 80 to 95%. However, as you move to more solid-like tumors like chronic lymphocytic leukemia, the complete remission rate remains around 20 to 30%. And why is that? Well, it has to do with some of the morphological features of chronic lymphocytic leukemia. See, in ALL, the cells were blasts. They're immature. But in CLL, the cells are actually more differentiated. They're kind of mature B cells, but they're dysfunctional. They're dysfunctional mature B cells. And if you take a look at this figure on the right, if the overall response rate, they had a lot of patients had partial response, but the amount of them staying inside complete remission was quite low. And so complete remission is a term used when we don't detect, there's no minimal residual disease. Just because a patient's in complete remission, this does not indicate that they're tumor free. We just can't detect the tumors at that time. And so one of the reasons why these patients are having not having very high complete remission rates is the tumor keeps relapsing. And when they relapse, they have an antigen negative relapse. So after they undergo these CD19 CAR-T therapies, they relapse and the CD19 antigen is gone. So there's a couple ways that this can happen. Number one is just alternative splicing, right? So in this figure A here, normally when if the protein is spliced correctly, you're able to detect CD19 epitope on the extracellular domain. However, alternative splicing, you lose the epitope and you're not able to target them with CD19 CAR-T therapy anymore. Or in panel B, if you're putting such immune pressure on these blast cells that have CD19, remember, you may have minimal residual disease that you cannot detect any cancer. But after a few years, those B cells without CD19 may relapse, rendering any previous treatment not able to function again in the future. Or if you can have there's been some cases of where you put immune pressure on these AL cells that they do lineage switching. So in this picture here, they switch from acute lymphoblastic leukemia to acute myeloid leukemia, a cancer of the myeloid lineage of the hematopoiesis pathway. And there's been some cases of trogocytosis, which when cells are in close contact with each other, they're able to exchange some proteins on their membrane. So in this case, the CD19 antigen was exchanged onto a B cell or a T cell, leading to cooperative killing of your T cells, leading to the relapse of the blast cells. So CAR-T therapy is a last line treatment, but if your last line treatment fails, you're not really left with much other options. So how can we address this clinical issue? Well, we can address this clinical issue by new novel therapeutics. So in our lab, we identified another target to treat patients who failed CD19 CAR-T therapy. And this alternative target is to treat chronic lymphocytic leukemia. This target is called B-cell activating factor receptor. Now, if you take a look at this picture, CD19 is present all the way from the pro-B cell all the way to the plasmablast. However, like we said earlier, when the patients relapse, CD19 antigen is lost, leading to no more CAR-T treatment for these patients. However, BAFFR, it's a good target because it's also expressed on immature B cells all the way to the plasmablast. However, it's a good target because it's specific to B cells, right? And remember, CLL is mature dysfunctional B cells, not blast cells. It's also not present on your hematopoietic stem cells. And unlike CD19, BAFFR, we don't know if it can be downregulated. BAFFR plays a key role in the survival and development of the B cell. So downregulating this in a B cell is most likely going to lead to death of that B cell. So we don't know if it can be downregulated like CD19. And previous studies have shown that CLL expression is quite high or BAFFR expression is quite high in CLL. So now we're going to talk about our CAR-T design. We have a second generation CAR-T design. So we have a one costimulatory domain. So we have our antigen recognition domain, your BAFFR single chain variable fragment, a hinge, which provides flexibility to the CAR. We use a CD28 costimulatory domain to increase the proliferation and expansion of the CAR-T cells, the CD3 zeta intracellular activation domain, and then we add an extra gene called truncated EGFR. So truncated EGFR does a couple of things. It does two things, actually. So after we introduce our lentiviral vector to make our CAR-T cells, truncated EGFR will be on our CAR-T cells, and we can use this to quantify the amount of T cells that are actually turned into CAR-T cells. And the second way is the more clinically relevant way is we can use when we infuse our CAR-T cells into patients, and let's say they have adverse events. We could administer cetuximab, which is the anti-EGFR antibody, which will shut down the CAR-T cells in the case of any adverse events. So this is why we include truncated EGFR into our construct. But now we'll be talking about the CAR-T production overview. So how do we make CAR-T cells? It usually involves 5 steps. First step is to identify a target antigen, right? One that's ideally present on your tumor, but not on your healthy tissue. You make your retrovirus CAR gene, and then you can put it into a viral vector and use this virus to deliver your CAR gene to the T cell. You do preclinical assay to prove that your CAR-T cells are only targeting the cells you want and not the healthy tissue. And then you move on to a phase 1 clinical trial, and eventually phase 2 and phase 3 until they're FDA approved. So we'll talk a little bit briefly about each one of these steps. First, we're going to talk about step 1. We've already identified that BAFFR is a good target for chronic lymphocytic leukemia, but now we need to produce the antibody that targets BAFFR. So we do that by using what we call hybridoma technology. Now, this is a process of immunizing a mice with your target antigen that you want to create an antibody for. So in this case, we wanted to create a BAFFR antibody. So we immunized the mice with a cell line that's overexpressing, a murine cell line that's overexpressing BAFF receptor, with the hope that the mice will have use this as an immunogen, create an immune response, and make monoclonal antibodies against BAFF receptor. And so after a few weeks after immunizing the mice, we will harvest these B cells from their spleen. And the problem is that these spleens, B cells, only survive in culture for about 21 days or 28 days. So we have to fuse them with a myeloma immortal cell line to create what we call a hybridoma. And with these hybridomas, some B cells may not fuse with other melanoma cell lines. They may fuse with B cells. B cells may fuse with B cells. B cells may not fuse. And so we have to select it out using ELISA. We use a selective medium called HAT media, which selects out for the hybridoma clones. Once we have these antibody-producing hybridoma clones, we test their antibodies that they make and see if they're specific to the BAFF receptor. And once we have our antibody of interest, and we'll start identifying a lot of different hybridomas that make this BAFF receptor antibody. From there, we stock up on these antibodies, and we will then test to see which antibody has the highest specificity and affinity for BAFF receptor. From there, we'll sequence out the heavy and light chain and subclone that into a lentiviral CAR-T plasmid. This is the first step of CAR-T production. Any CAR-T therapy has to undergo antibody production and antibody affinity and specificity screening, as well as subcloning into a retroviral vector. Now we're going to move on to using this retroviral vector. How can we use a retroviral vector to deliver our CAR gene into T cells? So to understand that, you need to know a little bit about the HIV structure and genome, as well as their replication cycle. So let's talk a little bit about their structure and genome. So in the HIV genome, you normally have 9 genes: GAG, POL, envelope, TAT, REV, NEF, BIF, BPR, and BPU. And they're flanked by these long terminal repeats. These long terminal repeats serve as instructions that anything in between these 2 long terminal repeats are going to be integrated into the host genome. And as you can see, each of these proteins or these genes encode for specific proteins. So POL encodes for your protease, reverse transcriptase, your integrase. Your GAG encodes for your structural genes, such as your matrix and nucleocapsids. And one gene I want you to focus on particularly is in the envelope gene, we have a surface protein called GP120. This is what the virus normally uses to bind to CD4, right? So now we have to look at the replication cycle. It usually encodes for 6 steps of how a retrovirus infects this target cell. It will bind to this target cell, right? GP120 binds to CD4. So it targets CD4 T cells. It will uncoat its nucleocapsid. and reverse transcribe its DNA or RNA into DNA. It will integrate into the host genome, and it will make RNA molecules to make proteins, and as well as new genome. It will assemble and bud from the cell to make new retroviral variants. However, we don't want that. We only want steps 1 through 4. We wanted it to integrate into the host genome, and we want it to stop. So how does it go through doing that? Well, we use what we call a recombinant lentivirus system. So again, this is a normal HIV genome. It has these long terminal repeats here, and these long terminal repeats contains these 9 genes. So we're going to manipulate it in a way that it will only integrate into the host without protein assembly and synthesis. So what we did was we split up what we call a 3rd generation lentivirus system, where we had, let's go through it step by step. We have a self-inactivating vector. This is our first vector here. And you can see here, these long-term repeats. Remember, this tells the virus integrate everything into between these 2 lines into the host genome. We only include our transgene of interest. In this context, it would be our CAR-T genes. And then we have a packaging vector, which is a separate vector. There's no long-term repeats, so these will never be integrated into the host genome, but we need them to make a structural and functional retrovirus. So these encode for your GAG and POL genes, as well as your REV. And then we have an envelope vector. Remember earlier I said that these retroviruses, usually the envelope protein is GP120. Well, scientists have changed it to VSVG. Now, VSVG is called the vesicular stomatitis virus G protein, which is put in place of GP120 to increase viral tropism. So instead of this virus only being able to infect CD4 T cells, it can now infect any cell that has low density lipoprotein. So you can now infect any cell that has a plasma membrane. And then to put it all together, just a quick schematic, we have your transfer vector with your cDNA, your envelope vector with your envelope protein, and your packaging vector, which contains the structural genes necessary for a retrovirus, like reverse transcriptase. And you put it into a packaging cell line. It'll make those proteins. However, its genome, right, that is going to integrate into the host will only contain your cDNA. So this is how we're able to harness the power of the retrovirus and use it to deliver gene as a gene delivery system. All right, retroviral genes are not present in this retrovirus. It's only our cDNA of interest. And again, just to reiterate again, this is just another schematic. You have 3 plasmids, and you have your transfer plasmid here with your transgene of interest. It's going to use this host machinery to make the viral proteins, to make a functional retrovirus. And then in green, denoted in green here, it only contains your transgene. And after it integrates into the host genome, only the transgene is there. And there's no more GAG and POL that gets integrated. So the retrovirus will never be able to replicate another replication cycle. Okay. And because CAR-T cells, this whole process takes about 14 days from isolation of PBMC from patients' bloods to making a functional CAR-T cell. It takes about 14 days. And throughout this whole 14 days, we undergo very strict quality control and quality assurance. So we have certain criteria that all our CAR-T batches need to undergo. So, for example, they need to expand to a certain fold. They need to have at least 25 expansion fold by day 14. These T cells need to be viable. They need to be alive. Their identity, right? Our population of cells, they need to be T cells. They can't be B cells, macrophages. And so we measure them by CD3 expression. And then we measure their potency. We use this, like earlier, we have a truncated EGFR. Potency basically means if you have a million T cells, how many of them are actually CAR T cells? And we identify this by using EGFR. And then we have WPRE, which is the RNA enhancer element. It helps the CAR get exported from the nucleus into fully functional protein. And then we have that VSVG protein we were talking about. We don't want to see this because this is a viral peptide. So we want to see less than 2.5 copies per 50 nanograms of DNA. And we do this using quantitative reverse transcriptase PCR. So let's go into some of our preclinical data now. Now that we know how CAR-Ts are made, how we use retrovirus, let's see how we test our CAR-T cells for cytotoxicity. We use two assays. We use a degranulation assay and a granzyme B ELISA. So if you take a look at this figure here, this is your inside all your T cells. You have these cytolytic granules, and in these granules, they're membrane-bound. They're membrane lysosomes that has these granule cores, and they have porphyrin, granzymes, granulysins, and proteoglycans inside there. But specific to the membrane of these lysosomes, we have this marker called CD107 alpha, which is also known as LAMP1. This is called the lysosome-associated protein 1. And upon activation of your T cell, you have mobilization of these granules. These mobilization will cause these granules to fuse with the plasma membrane, therefore allowing you to start to measure the release of granzymes by ELISA, and measuring surface expression of CD107 alpha only on activated T cells. If a T cell is not activated, you won't be able to measure CD107 alpha. And this is another schematic. This is actually an NK cell. They also degranulate. CD107 alpha right here, denoted in purple. When they're mobilized and fused with the membrane, you're able to detect the presence of CD107 alpha. So you get to know when the cell degranulates, and you can also measure the amount of granzyme or porphyrin that was released to the environment. So this is how we tell that our T cells are activated. So now we're going to move on to the data, but before we can move on, we have a lot of flow cytometry data. So you need to understand how to read a flow cytometry graph. So this is your typical flow cytometry graph, and it's usually divided into 4 quadrants if you're measuring two surface markers. So if you take a look at this x-axis, we're measuring for CD3 expression on a certain cell. A shift to the right in quadrant 3, this means the cell is CD3 positive. However, they don't have expression for CD19. If you're looking at cells in quadrant 2, this upper right quadrant, that means they have expression for both CD19 and CD3. If you look at the y-axis, if there's a shift of population of cells in the top left quadrant, that means they are positive for CD19 cells, but they don't have CD3 expression. And a quadrant in the bottom left indicates a double negative. They don't have any surface expression of both markers. Okay, so how can we prove that our CAR-T cells target only our cancer cells and not healthy tissue? Well, we need to use different cell lines. So in our studies, we use 3 cell lines. We use a acute lymphoblastic leukemia cell line, mantle cell lymphoma or non-Hodgkin lymphoma, and a chronic lymphocytic leukemia cell line. And so we use these 3 cell lines, and we wanted to test if the BAFFR expression is high in all 3 of these cell lines. So this panel right here, we're seeing that this FMO live is our negative control. All 3 cell lines have a shift of BAFFR to the right, meaning they have high amounts of BAFFR expression. Okay, now let's go ahead and mimic the clinical setting, and let's knock out CD19. And while we're knocking out CD19, let's make sure that our BAFFR expression is not affected. So we take a look at this top panel here, this NOM6 cell line, which is our ALL cell line. We'll start with this one. In the wild type, you can see that there's BAFFR expression in this top panel. And you look down at the panel below, you can see that there is CD19 expression. There's a shift to the right, okay? Let's go ahead and knock out CD19, and let's see if it affected our BAFFR expression. So CD19 is knocked out, right? There's a peak where the red peak is, but BAFFR expression is still there. So this is a model we can use. Then when you look at your non-Hodgkin lymphoma cell line, there is a decreased expression of BAFFR compared to the wild type, but there is still BAFFR expression, and the CD19 is knocked out. Same with this one. This one, there's actually, it looks like there's an increase in expression of BAFFR after knocking out CD19. So these are the cell lines that we'll be using to test our CAR-T cells. So let's look at our data. So this is our degranulation data as well as our granzyme B ELISA. So we take a look at panel A. We gated CD107 on CD4 T cells. So these are only our CD4 T cells. And MC1029, this is just another nickname we gave to our BAFFR CAR-T cells. So this is the treatment group. Non-CAR T cells, these are T cells that have not been introduced to the retrovirus. So they do not have a CAR gene. They're just regular T cells. So when we co-culture these non-CAR T cells with our NOM6 wild type, we'd have no measures of CD107 alpha. There's no expression of CD107 alpha. And we see the same phenomenon when we knock out BAFFR. There's none. However, when we co-cultured our BAFFR CAR-T cells with a NOM6 wild type, which is BAFFR positive, we were able to measure expression of CD107 alpha on the surface. However, whenever we do that same experiment using our BAFFR CAR-T cell co-cultured with a BAFFR knockout cell line, we do not see any expression of CD107 alpha. So this is telling us that our T cells, our CAR-T cells, are only activating in the presence of their specific antigen. So they have what we call antigen-specific degranulation. Now, do we exhibit this effect in both CD4 and CD8 cells? We do. So if you take a look at panel B, this is gated only on your CD8 CAR-T cells. We can see the same phenomenon. When BAFFR is knocked out, there is no degranulation. And as expected, right, CD8 T cells are our cytotoxic T cells. There are larger events of CD107 alpha on the surface of these T cells. And panel C is our degranulation measuring of granzyme B. Because CD107 alpha is directly correlated with the amount of granules being released to the environment, we see what we should expect. When BAFFR CAR-T cell, MC1029, is co-cultured with a NOM6 wild type, you see an increasing expression of granzyme B. However, when BAFFR is knocked out, we see no granzyme B expression in the environment. Now let's do a more direct killing approach. Let's label our NOM6 ALL cell lines with green fluorescent protein. And then let's co-culture them with our CAR-T cells, and do we see any change in expression of green fluorescent protein? So in panel D here, this is our normal T cells, our non-CAR T cells, expressed with NOM6 wild type, and you see about 4% expression of green fluorescent protein. Now let's introduce our CAR-T cells to these NOM6 wild type. What happens to the green fluorescent protein? There is a significant decrease in green fluorescent protein, indicating that these CAR-T cells are killing these ALL blast cells. It went from 4% to 0.086. And then the same phenomenon, if we knock out BAFFR, there is no decrease in green fluorescent protein, showing, again, the killing is antigen specific, which is what we want to see. Now let's move on to animal models. Can we see the same phenomenon in animals, right? So we use our NOM6 cell line, our ALL cell line, into animals. So on day one, we engrafted these animals with tumor cells. So this would be our NOM6 ALL, and we transfected them with luciferase. So a higher fluorescent intensity indicates a higher tumor burden. So these mice here, we had three groups. We had a PBS group, a normal T cell group, and our treatment group. And as you can see in this plot over here, these mice in the saline group and the normal T cell group, they all succumbed to disease and died around day 60 and 45 or 50. However, in the treatment group, these CAR-T cells were able to promote tumor-free survival in these mice. And this is our Kaplan-Meier plot, showing the survival curve of these mice. And green indicates the CAR-T cells treatment group. Okay, now let's do the same experiment again, but let's knock out CD19 and use CD19 CAR-T cells to show what's really going on in the clinic. So in panel A, we had a CD19 knockout model instead of a BAFFR knockout model this time. And when we use T cells against normal T cells versus a CD19 knockout model, there's no degranulation events. We don't measure any surface expressions of CD107 alpha. And when we use our BAFFR CAR-T cells, we measure a 26% surface expression of CD107 alpha. And when we co-cultured, as we expected, when we co-cultured a CD19 targeting CAR-T cell with a CD19 knockout model, what we see in the clinical setting, we do not see any surface expression of CD107 alpha, meaning that these cells are not activating because there needs to be that specific antigen there to activate it. And BAFFR can still activate even in the absence of CD19. And again, our granzyme B data is consistent with our CD107 alpha expression. Can we see the same effects in a vivo model? So we had a CD19 knockout model, and you can see this time we had 4 groups. We had a CD19 CAR treatment group, a BAFFR treatment group, a T-cell treatment group, and a PBS treatment group. As you can see, in the PBS treatment group and the normal T-cell groups, the mice eventually succumb to disease burden. However, in the BAFFR group, the mice, again, they had tumor-free survival. And the CD19 treatment group was quite interesting. The mice followed the same disease burden as your PBS group because the antigen wasn't present, but we had one odd mice that had tumor burden, went into remission, and relapsed again on day 84, but eventually died to tumor burden. Okay, so this was our treatment in acute lymphoblastic leukemia. Can we see the same effect in lymphoma? So we use our other cell line, Z138, and we wanted to test, is it also antigen specific in a different cancer? So this is lymphoma, and as you can see here, gated on CD8 CAR-T cells. Again, our BAFFR is only degranulation in the presence of the antigen. Normal T cells will not degranulate. They need that CAR expression to degranulate. And the granzyme B data is consistent with our CD107 alpha. Can we see the same effect in vivo models? And indeed, even in the lymphoma model, we see the same phenomenon. Our BAFFR CAR-T cells are able to induce tumor-free survival in lymphoma in mice. However, we did have one mice relapse from the disease. It was tumor-free for on day around 60, and around day 80 or 75, it started to relapse, and we were able to detect the cancer again. And this could be due to a various number of reasons. It could be that our T cells stopped proliferating. It could be perhaps the BAFFR expression got downregulated. We don't know. There's more studies that need to be done. And finally, our target disease of interest, chronic lymphocytic leukemia. This is where things get a little interesting. So again, these BAFFR CAR-T cells are showing this antigen-specific cytotoxicity. They're only degranulating in the presence of the target antigen, okay? So let's move on. This, in our studies with CLL, this is where we start incorporating primary patient tumors. So we had a doctor at Mayo Clinic, Dr. Carfon. He was a hematologist, oncologist. He was able to provide us patient samples, primary patient samples, bloods of patients with chronic lymphocytic leukemia, B-cell chronic lymphocytic leukemia. And as you can see, we got 6 patient samples here. And the demographics of these patients are all elderly individuals. And what we did was we extracted 50 ml of these patients' blood, isolated their PBMC, and further even isolated out only their B cells. So we had primary patient tumors from patients. And as you can see, after we extracted B cells from 6 of these patients, we wanted to measure to make sure that they have BAFFR expression. And as you can see, all 6 patients had a significant amount of BAFFR expression in their tumor cells. Okay. Now, if we co-cultured healthy T cells with these primary patient tumors, and we're hoping to see cytotoxicity. Can our T cells kill these primary patient tumors? And what we saw is we had T cells from 2 donors, donor A and donor B. We had T cells from that were manufactured from donor A and T cells manufactured from donor B. And what we saw was that in patient samples, our CAR-T cells are activating only in the presence of BAFFR. When we use normal T cells against patient samples, we do not see any degranulation events. And the same with when we quantify the amount of granzyme B released into the environment. Only in the presence of the antigen can our CAR-T cells release their granzyme B into the environment. And then it's important to note that most CAR-T clinical trial studies now they use autologous CAR-T cells, meaning that the T cells that they use to manufacture into CAR-T cells, they always come from the patient. So the reason we don't do allogenic is because a phenomenon called graft versus host disease, right? We only want graft versus tumor. And so we wanted to mimic that by additionally getting an additional 3 more patients with B-cell chronic lymphocytic leukemia. And we grabbed 50 ml of blood, we spun it down by density gradient centrifugation, isolated out their PBMC cells. From there, we further isolated out their B cells. From that same PBMC, we further isolated out their T cells, introduced the retroviral particle, and turned these T cells into CAR-T cells. And we co-cultured their own patient's T cells with their own primary tumors, just like in the clinical setting. And we wanted to see if these CAR-T cells were healthy enough or fit enough to kill their own tumor cells from patients. And this is just one of the quality assurance, quality control assays that we did. And so in the first panel here, we measured the identity of patients' T cells. So are they CD3 positive? Are they actually T cells, right? As you can see here in subject 7, 8, and 9, they had 99% of the sample that we measured had CD3 expression, meaning they are T cells. Now, how much of them, what's the potency? How many of them expressed EGFR? Remember, EGFR is the expression for CAR-T cells. So of this patient population of T cells, of subject 7, 23% of the T cells we transduced with the virus are now CAR-T cells. 21% are now CAR-T cells for subject 8, and 34% are CAR-T cells for subject 9. So let's go ahead and see the degranulation data and let's see what we see. So let's take a look at subject 7 here. When we perform this assay, when we perform B-cell co-cultured with their own T cells, CAR-T cells, we see a degranulation event about 3-folds compared to the normal T cells. And we use a MEK1 as a control. And in subject 8, we can see almost a 5-6-fold increase of degranulation events when co-cultured with their own primary B cells with their own CAR-T cells. And same with subject 9. In subject 9, we saw a 10-fold increase in degranulation events. So what can we get from this? That patient CAR-T cells can kill their own tumor B cells. These are autologous derived CAR-T cells. And then finally, the last step of it all, moving on to a phase 1 clinical trial. So after all that, we can finally move on to test to see if our CAR-T is safe in human subjects. So this will be the first CAR-T therapy to develop at Mayo Clinic and to be manufactured on the same site. So this is quite literally the philosophy of bench to bedside. We are doing research in the bench and we're bringing it directly to the patient's bedside. And there's a couple, being able to manufacture it and deliver it at the same hospital brings a couple advantages, right? We're able to adjust doses on the fly, individualize patients' treatments, and deliver the cells right back to the patients in a promptly manner. And this is our new discovery and innovation building in Florida. This is where we'll be biomanufacturing our good CAR-T cells in GMP conditions, good manufacturing practices. And these patients will be housed in the ICU, and we will deliver these CAR-T cells directly to them. And so this data here is just showing that we can indeed produce CAR-T cells under GMP conditions. So we meet all the expectation thresholds. The cells expanded properly. They're alive, 9488, 93% of the cells are alive. 98% of the cells are actually CD3 positive. They have very high CAR gene expression. And we show that the virus is replication deficient. We don't detect any copies of VSVG, very low traces. So in conclusion, we exhibited that MC1029 or BAFFR CAR-T cells exhibited antigen-specific cytotoxicity in acute lymphoblastic leukemia, chronic lymphocytic leukemia, and lymphoma, both in vitro and in vivo. CAR-T cells remain effective models. Our BAFFR CAR-T cells remain effective even in the absence of CD19 antigen loss, which is what we see in the clinical setting. Patient-derived autologous BAFFR CAR-T cells are able to eradicate primary patient tumors in vitro. And currently, we're ongoing to that phase 1 clinical trial in patients, and we hope to have this drug FDA approved by the year 2030. And some acknowledgements, my mentor, Dr. Hong Chen, right here, MD, PhD, and also Yan Luo, they both did their MD, PhD in China, and they were great mentors to me who I wouldn't be where I am today without these two mentors, and then all the other lab members. And special thanks to MD Anderson Cancer Center in Texas for helping us produce our monoclonal antibodies, as well as the Animal Resource Center at Mayo Clinic for their daily care of mice used in this study. And here's a list of my references, and then we can open up for questions.
Excellent, excellent. Tommy, that was amazing and validated one of my long-held beliefs that many aspects of virology blended with immunology, biochemistry, molecular biology, pretty much everything can eventually be traced back to the study of viruses.
Yep.
And that it's been harvested, that knowledge, and used to cure really devastating forms of blood cancer that were previously more or less untreatable is just a stunning development. So I've a couple of comments. Number one, what a great talk. It's the best outline of CAR-T that I've seen, way down to the nuts and bolts. And it's clearly a team sport. So what was your role in all of this? What was your specific role?
Yeah. I played a role in the cytotoxicity assays, as well as generating these CAR-T cells. So I played a role in cloning the heavy and light chains of the CAR-Ts into a lentiviral vector, into the production of the lentivirus, into the transduction of the CAR-T cells, and then as well as testing these CAR-T cells in the animal models. So I played a role in the in vivo studies as well as the in vitro data. So I helped, I guess, and you could say in all phases of the steps.
My goodness, what a stunning opportunity for such a young man. Great accomplishment. I see Dr. Johnson has a question. Go ahead, Mike.
Tommy, I can't echo enough in terms of praise to your presentation. Dr. Steininger said it was awesome, and that's times 10 from my perspective.
Thank you so much.
Well presented. I'm going to ask you, you played such a vital role in this, and very humble presentation as you're making this. When I ask you, what part of this this area that you were involved in surprised you the most, that you didn't see coming, or you didn't anticipate, or perhaps looking back and say, gosh, this is better than we thought?
Yeah.
What area was the most surprise to you in this study?
The area that most surprised me was when I was shadowing Dr. Carfon in the clinic. I wanted to see, I asked him, could I see some of the side effects of patients when they receive CAR-T cells?
Yeah.
And some of the things I saw the most was that they had a lot of cytokine release syndrome, and they had a lot of ICANS, they had a lot of neurotoxicities, but we were able to medically treat them, because one of the questions I had for Dr. Hong Chen, my mentor, was that CD19 is a target that's on all of our healthy B cells and our disease B cells. Same with BAFFR. They don't differentiate from disease cells as well as healthy cells. So how can we target this, and how can the patient still be alive if we just have complete B cell aplasia? And they told me that in the clinical setting, complete B cell aplasia, it can be medically treated. That was one of the things that surprised me the most. And another thing that very surprised me is one time we had a patient that he had chronic lymphocytic leukemia, and their blast cells, their immune cells are overcrowding their bone marrow. So sometimes you cannot get healthy enough T cells to manufacture into CAR-T cells. And so that patient had to undergo an allogenic CAR-T transplant. And what we saw in a couple months, 6-7 months from there, we saw graft versus host disease in this patient. And that was also a very, something that very surprised me. Graft versus host disease is really devastating in patients, and there needs to be ongoing research to address this issue.
Okay, again, thank you very much. It's just an outstanding presentation.
Thank you so much.
I see Dr. Wolf has a question.
Yeah, sorry, I got here late. I will echo that I thought it was an outstanding presentation, what I saw. You touched a little bit on what I was curious about, and that was the lack of specificity. And so I was curious, I got here late enough that I didn't necessarily see what payloads you took, but I'm thinking, well, if my only goal is to blast B cells, why don't I just come in there with an antibody that's going to activate complement and destroy the whole lot of them, rather than going through all this CAR-T stuff?
Well, that's a great question. I think that is certainly an option, but I think that when you're dealing with the patient, you have to look at all options. I'm not sure if you blast with an antibody that activates a complement and degrades all the blast cells, could this affect other immune cells? Could this affect any other adverse events?
Well, I mean, I was thinking along this. I mean, so I didn't know BAFFR off the top of my head, because I don't think about immunology, but it says it's on, all B cell lineages or something like that. Or I mean, you're sitting there, you're looking at CD20 rituximab for certain things, and this struck me as being something that would potentially be broader scale, and so I was struck with, what good does this do you if you can't distinguish between cancer cells and non-cancer cells? I mean, so for CLL, if you've got a specific monoclonal production going on there, is it instead possible to somehow tailor that specifically to that population of cells based on what they presumably would have in their HLA, on their cell surface?
I wouldn't believe it's possible, because BAFFR, like you said, is even expressed on your healthy tissue cells. And that's part of the problem with CAR-T therapy is, let me go back a couple slides, is identifying a target antigen that's present only on your tumor cells. And part of the reason why that's such a hard thing is these tumor cells, right, were once healthy cells. They just got mutations and they eventually accumulated into cancer cells. So this first step is very crucial. Identifying a target antigen that's present only on cancer is very tough. And this is part of the reason why, if you take a look at this slide, as you move into...
I mean, this is way before I got here. So I apologize for kind of
problem.
asking about stuff you probably had gone through.
So in our lab, we actually tried to target EGFR V3. So this is a antigen that's found on glioblastoma, a brain cancer, but it's very hard for CAR-T therapy to function because of the tumor microenvironment, how harsh it is for T cells to survive there. Just to traffic the T cell there, we have to inject it locally. And EGFR, it's very heterogeneous. Not all the tumor cells express it. So identifying a target antigen that's ideally present only on your cancer cells is very not very feasible. I would say it's very tough.
Yeah, so actually I came in when you were talking about modifying to target the LDL receptor, and I heard that part, and I'm going, well, how are you going to keep it off of the liver? And so, but so you're separating out the blood, so you're not giving it systemically. But then as I thought a little bit more, I mean, are you confident that you've eliminated all the virus? I mean, are you actually carrying some of that bound to your T cells in, and is there expression in systemic tissues based on the ubiquity of its binding?
Right. Well, based on the I would say I'm pretty confident that the CAR-T cells, the retrovirus, wouldn't be able to replicate again, because when we're integrating the genes there, like I mentioned earlier, that the retroviral structural genes needed for replications will never be integrated. It's only your transgene of interest. And so once
I understood that, but I was going, if you got the LDL receptor on the surface, do you have virus going along as adherent cargo?
Oh, okay. That's a great question. That's great. I never really thought of it from that angle. It could be a possibility. I'm not really sure how we could test for that.
And I don't necessarily know that it's that important, but that was one of those things that I'm going, okay, where, you know, the next step, I mean, because once you put that virus in, you would conceivably be able to exert effects on any cell that expressed that protein and, or, you know, the LDL receptor, and clearly the body's got a lot of them.
Yeah. Yeah, it's amazing what the retrovirus can do, how we can use viruses for therapeutics.
So I have a basic question for you, Tommy.
Yes.
CAR-T cell therapy has been used successfully in a number of different cancers. Please tease out in very basic terms, what's the novelty of this approach compared with this, you know, what's become the standard of care in CAR-T cell? I realize it's pretty new, but what's novel about this?
Well, let me go back to the structure of our design. So based on our design, what's really novel about it is that we added a kill switch. So one of the adverse events of infusing millions of T cells in the body is when they bind to the target, they release a lot of cytokines, such as IL-6. Well, one of the novelties we have in our design is we introduced the kill switch. The truncated EGFR, we could use cetuximab to turn off the T cells in the events of any cytokine release syndrome or any adverse events that we were not expecting. So this is one novelty that's in our design.
So that's pure genius. And have you seen that function in mice yet? Have you killed it in mice using?
Yes, we tested it, but we didn't publish it, but we tested if we can see that if we introduce cetuximab into a mice model, will it turn off the CAR-T cells? And indeed, they do turn off the CAR-T cells. And
That's amazing.
Yeah,
that's amazing. Again, you know, harnessing the tools of immunology, biochemistry, molecular biology, all the way to patient care, bench to bedside. Such a stunning example of what can be done by incredibly smart and ambitious people.
Yeah.
And for you to be part of that team, just amazing.
Dr. Hong Chen was one of my greatest mentors. And, you know, one quote that he stuck with me, that still sticks with me, is he told me when I become a physician one day, and if I do go through the hematology oncology route, he said, you will never remember the patients you save. It's always the patients that you didn't get to save that will stick with you throughout the years. And he was telling me about when he was in medical school, his first patient that died on him stuck with him still to this day. So, and that's why he does all the research he does. So it's quite humbling.
Yeah, Mayo, high level stuff. Hopefully, you know, you achieve your dreams and land in a Fred Hutchinson, MD Anderson, Mayo type place. My goodness, that'd be so fun and rewarding. That's a career worth fighting for, my friend.
I do want to become a physician scientist. I feel like medicine has too many limitations for us to stop moving and advancing forward, Dr. Seldinger.
Yeah.
Could I ask real fast, how long did those cells live?
The CAR-T cells?
Yeah.
They usually circulate depending on if let's say if the CAR-T is circulating throughout the body and they're not being activated, they would die in about 3 to 4 months. But if the tumor cells still there and they're constantly being activated and expanding, they will live for years, 6 months to years. Yeah.
Okay.
Depends on whether they get continuous activation.
So does then the number of virus particles, or not virus particles, CAR-T cells that you infuse back into the patient, that's probably an amazingly critical aspect of treating humans.
Yes.
Is that right?
We determine that based on your weight. So there's a certain formula that they do in the clinical setting where you weigh the patient and then you divide that by a certain factor and then you calculate how much CAR-T based on the patient's body weight.
That's great. Okay, any other questions for this high-level operator here. Let me thank you once again, Tommy. Look forward to mentoring you in medical school, if at all possible, at KCU. I wish you Godspeed in your journey, my friend. That was stunning. Thank you so much for your
Thank you so much for your time.
You bet.
Thank you.